So the cell has two types of endoplasmic reticulum, rough and smooth. The rough endoplasmic reticulum is so called because it looks rough down the microscope as it is covered in ribosomes. And the smooth is so called because it is, well, smooth. The endoplasmic reticulum is a convoluted single internal membrane that has an internal space that is referred to as the ER lumen. The smooth ER lack ribosomes and is the site of vesicle formation to transport proteins and lipids to the Golgi apparatus for further processing. The rough ER, as I have said, is coated in ribosomes, and it is here that protein production occurs. Protein production starts in the cytosol, and as the N-terminal emerges from the ribosome messenger RNA protein complex, it is sampled for a signaling motif by a protein called the signal recognition particle, SRP. If the signal is present, then the SRP binds, stops translation and directs the protein ribosome messenger RNA complex to a receptor on the endoplasmic reticulum. And this makes the rough ER rough. The protein is then made co-translationally. That is, protein production restarts once the ribosome is bound to the ER and the protein, as it is made, is passed through a pore, the protein translocator, Sec61 complex, into the lumen of the ER. You should note that the protein may be passed as one molecule into the lumen of the ER, or it may be membrane spanning. So how does a membrane spanning protein get made? Now, we had proteins that span the membrane once or multiple times. Some proteins have the C-terminal outside the cell and some inside. How can this be achieved? Well, there are a number of different types of signal that can be encoded in the amino acid sequence. There is the N-terminal signal, which in this case targets the protein to the ER. And there are also internal stop transfer sequences that stop the threading of a protein across the membrane. And this type of signal results in the formation of membrane spanning domains. And this will produce a protein that spans the membrane just once. However, there is also an internal signal sequence which is similar to the N-terminal signal, and if one of these sequences occurs after the stop transfer sequence, then threading into the ER lumen will restart. Hence, through a combination of stop transfer sequences and internal signal sequences, it means that proteins can be threaded backwards and forwards across the membrane as required. Finally, two other functions of the ER that are worth noting. First, if the protein needs to be glycosylated by the addition of an N-linked oligosaccharide, then this will happen as the protein is threaded into the ER. This will then be followed by some further processing in the ER and completion in the Golgi. Such proteins are called glycoproteins, and in this case N-linked glycoproteins. Second, the ER is the gatekeeper of protein quality. That is, the ER checks that the proteins are correctly folded and will degrade any proteins that are not.